Okay, so this is the sarcolemma. It is a barrier between the extracellular and intracellular components. And then this is the sarcoplasmic reticulum, and that stores calcium ions and regulates the calcium ion concentration. And then this is the T-tubule, and that conducts impulses from the sarcolemma into the sarcoplasmic reticulum, regulates calcium concentrations, and permits rapid transmission of the action potential. And then down here we have the sarcomere, which is a contractile unit of a myofibril that shortens by the sliding of the thick and thin filaments.
Alright, so this is how a muscle contracts. There's an action potential that goes down a motor neuron. This action potential allows for a voltage-gated protein to open up and calcium ions go in. The calcium ions allow for the vesicles containing acetylcholine to move down the synaptic knob. Exocytosis occurs, which lets out the acetylcholine into the synaptic cleft. This is a process that utilizes ATP. Next, there's an influx of sodium ions that go in through these channel proteins, and potassium goes out. But then in order to create a concentration gradient, the Na needs to be pumped back out and potassium needs to be pumped back in. This creates another action potential that goes down the T-tubule. When there's an action potential along the T-tubule, the voltage-gated proteins open up again and calcium pours out of the sarcoplasmic reticulum. And then then the calcium binds to the troponin-tropomyosin complex, which then moves the tropomyosin from the active site on the actin. The calcium, Then the myosin head binds to the actin active site, and then there's a power stroke, which then moves the fibers in towards the midline, the actin fibers in towards the midline, where the myosin fibers stay put, and then the sarcomere shortens, and then the muscle shortens altogether.
Go.
Okay, so then for a muscle to relax, there needs to be no motor stimulus, which means that there's no calcium flowing in, and then the acetylcholine will not be exocytoted into the synaptic cleft, which means that there will not be a sodium-potassium pump that creates the action potential on the T-tubule. So then that means that no calcium will be pumped out of the sarcoplasmic reticulum. So this calcium needs to be reabsorbed by the sarcoplasmic reticulum, which needs ATP. And then since no active sites will be open anymore, then the myosin heads cannot bind to the active site, and then there will be no more power strokes, which means that the muscle fiber will not shorten anymore.
So on our sarcomere, we have troponin bind to tropomyosin, which makes a complex, and they're on actin filaments. And then when calcium binds to this complex, then it opens up an active binding site. So then when ATP binds to myosin, it cocks the head, and then the actin and myosin connect, and that's what forms the cross bridge.
So the myosin head is bound to the actin because of ATP, which is then hydrolyzed into ADP and P. And when that is lost, the power stroke moves the actin towards the midline, where a new ATP would be able to be picked up and cock the myosin head again.
Okay, so myosin and actin are contractile proteins, and this purple or dark band is the myosin, and the red or light bands are the actin, and the myosin pulls in the actin, so the actin slides over the myosin, and that causes the muscle to shorten, but not the filaments. So troponin and tropomyosin, along with calcium, are regulatory proteins because the calcium binds to the troponin, and that causes the active sites on the actin to be exposed, and that regulates when the myosin head can attach to the active site on the actin.
ATP is used in muscle contraction at the exocytosis of acetylcholine to the ligand-gated sodium channels. If ATP isn't present, acetylcholine would not be able to exit the neuron to open up those channels. ATP is used next at the sodium-potassium pump to pump sodium out of the cell and potassium back into the cell. If ATP isn't present, sodium would not be able to be pumped back outside the cell, and potassium would not be able to be pumped back in. ATP is next used at the myosin heads for contraction. If ATP isn't present, the myosin heads wouldn't be able to bind to the actin, resulting in no contraction. ATP is used lastly at the sarcoplasmic reticulum, where calcium is pumped back into the sarcoplasmic reticulum after the action potential has stopped. If there's no ATP, the calcium will not be absorbed back into the sarcoplasmic reticulum.
In this situation, we'll show you how a muscle contracts after a person has just performed some warming up exercises.
So a person warming up allows for a trepe twitch, which means that a stimulus is present right after relaxation, which means that there's calcium in the sarcoplasmic reticulum and it's not absorbed as readily as the other contractions. And this allows for your muscles to be warmed up before you need to use hard or fast twitch.
This situation is showing how a muscle contraction is affected when a person is dehydrated.
When someone is dehydrated, they don't have enough electrolytes in their body. Electrolytes contain sodium and potassium. Because there aren't enough sodium and potassium in the body, then the sodium, potassium involved in the action potential aren't present, so the action potential is weak, and therefore the muscle reaction is weak.
Ugh.